But first, let's talk a little bit about ATAC-seq data, what that is. ATAC-seq stands for Assay for Transposase-accessible Chromatin with High Throughput Sequencing. This assay basically tries to detect the open chromatin regions in the genome. The protocol relies mainly on the cleavage enzyme TN5. The enzyme TN5 will come into the genome and recognize the open regions, cut out the fragments, and then tag them with sequencing adapters. As a result, we can do PCR amplification and sequence the fragments, typically with pair-end sequencing. After receiving the reads, we can align the reads to the genome, and as a result, we would have pile-up of reads in certain regions in the genome. The downstream step would be to quantify and to determine the significance of the read pile-ups in the genome. And this process is typically referred to as peak calling. And the peak regions means they are open, or they could be called as nucleosome-free regions. Currently, there are multiple tools out there to do peak calling for ATAC-seq data. On the screen right now, I'm showing you different classes of methods. They can be roughly divided into traditional statistical methods and supervised machine learning. In today's talk, I want to highlight 3 representatives of the methods. The first one is MAX2. MAX is probably the most popular peak calling tool out there right now. It was originally developed for ChIP-seq data. It counts reads with sliding windows and then try to model the read counts with Poisson distribution. The second one that I want to highlight today is ChromHMM ATAC. It relies on a hidden Markov model and was built specifically to model ATAC-seq data. It categorized reads into nucleosomal classes and based on the fragment lengths, and then model each read class with a statistical distribution. The last one that I want to touch on today is Lance Ultron, which is a newly developed supervised machine learning tool. It uses labels from human experts and to use convolution neural network-based method in conjunction with some other statistical testing to score for the peaks. So as you can see, MAX2 and ChromHMM ATAC are representatives of traditional statistical modeling. So they handle only a few number of parameters. And with Lance Ultron, it is supervised learning, so it relies on external class of labels. And there's one knowledge gap here that I want to point out, is that they do not integrate information from biological replicates when calling peaks. So how are we currently doing to integrate information from replicates? There are multiple approaches. People can either merge replicates before doing peak calling and call peaks on one merged file. We can also do post-hoc p-value ranking to potentially eliminate the false positive peaks. And the last approach, which is also the simplest one, is to take union or intersection of the peak sets. These approaches have a few different ways to be implemented, or the example tools that I'm pointing out here. And they all have really good advantages. For example, they all help reduce noise, or they are very flexible. They can be applied to any set of peaks that have some sort of ranking or p-values. And they are pretty easy to be implemented. However, the innate disadvantages of these methods is that they ignore natural biological variation between the replicates, or rely mainly on peaks called by individual replicate. And also with the last approach, there's no standard threshold of how many peaks to take intersection or the union. I'm highlighting ChIP-seq here. ChIP-seq is a recently developed tool to do post-hoc p-value ranking, and it has been shown to perform better than the other approaches. So with these knowledge gap keeping in mind, we set out to develop a framework that is unsupervised learning to code picks for ATAC-seq data that can integrate information from biological replicates. We want to do an unsupervised learning method so that we don't have to rely on external labels. And we want to do machine learning method also because it potentially can handle higher dimension than traditional statistical learning and may achieve higher or better performance based on the observation from supervised learning. And also, we want to integrate signal across replicates from the beginning simultaneously, and maybe it will be a better way of utilize replicate information. With that, we have developed a framework called Replicative Contrastive Learner, or RCL. This framework relies mainly on the idea of contrastive learning, which is to learn a low-dimension representation so that similar observations would be close together and would be farther away from the dissimilar observations. To put it, let's get an example. Traditionally, contrastive learning is often applied to images data. From one source image, one can slightly distort this source image into augmented data and use the shared information from the augmented data to try to make a prediction on the source image. And in our case of RCL, we treat one genomic region as our source and the observations from different replicates as augmented data. So the idea is that we have different views of the same region from different replicates. Our goal is to classify the candidate segments into two classes, peak and non-peak. And the framework contains two main steps, candidate segment selection and to classify the examples. So whether the peaks cluster together would depend on several factors. For example, the signal versus noise ratio, or how similar the peaks are across replicates, and whether the true state of the chromatin region is actually shared across replicates. And regarding the last factor, we made an assumption about that the true state is shared across replicate in one specific segment of as a candidate. So let's talk about candidate segment selection first. We use genome per base raw coverage data from all replicate, and we would keep the positions that have the raw coverage greater than a threshold T. So T is the mean coverage to retain a position, and this is a user input. And as a default, we estimate them from the genome data. We discard the ones that are too small or merge the ones in a certain distance. And we explain the 90 base pair choice in our paper that I would have a QR code later. And then after we merged and finished the pre-processing step, we would extend the region from the midpoint to obtain an alpha base pair vector of raw counts. So alpha is defaulted to be 1,000. Amongst these two inputs, I want to highlight the importance of T. T, when we decrease T, it means that we would include more candidate segments with lower coverage data in the model training process. As the output of this step, we have vectors of per-base raw coverage at candidate regions. We use these vectors to put through our unsupervised learning training process, and we want to optimize the combination of three losses so that those similar regions would be close to each other, and the ones corresponding to peaks would be further away from the ones corresponding to non-peak. And in today's talk, I want to highlight the replicate similarity loss and the class similarity loss. With replicate similarity loss, we want to learn a lower dimension representation of the inputs so that the same segments across replicates are close together. So the green ones correspond to segment 1. They would be close together, and they are further away from the red ones. And the second loss relies heavily on the assumption that we made that the true state is relevant. The true state is shared amongst all replicates. So therefore, as we compute a probability of a peak for every segment, we want to make sure that the same calls are made for the same segment across all replicates. And this would contribute to giving us a score or a probability so we know a segment is a peak or not, and then it also contributes to the separation between the two classes. To evaluate the performance of RCL against the existing methods, we use true, open, or closed segments from different resources. The true is in quotation mark because they are noisy and they are estimated with computational methods and from ChIP-seq data. So there are two sets of labels we use. One is genome annotation that contains positive and negative labels. And the second set that we use is transcription ChIP-seq data. In today's talk, I will only mention about the results obtained from the first set of labels. On the screen right now, I'm showing an example of how positive labels and negative labels look like. The positive labels oftentimes correspond to active transcription start site or the regulative elements that are marked by histone modification ChIP-seq data, and it can be open so that transcription factors can come in and regulate genes. And the negative labels oftentimes correspond to the more quiet regions in the genome, and the chromatins, the nucleosomes tend to be close up together. And there's one thing that I want to note is that the labels do not cover every possible position in the genome, and that would lead to another additional analysis downstream of us. We tested RCL on 4 different human cell lines and 1 mouse tissue data, and we see a consistent performance of RCL being better than the existing methods. As you can see on the screen, RCL with a different threshold of T corresponds to the different 2 lines of color blue right here. And you can see that RCL performs better than the existing methods. And as we lower the T, we got better F1 scores, suggesting that exposing the models to lower signal regions would help the model to distinguish better between the peak and non-peak regions. As I mentioned earlier, our label sets do not contain every possible position in the genome. Therefore, we also look at the possible functions of the open regions by associating the regions with the genes. The idea relies on the assumption that open regions in the genome should be meaningful and they should be associated to functionally relevant genes. And we carry out analysis of peaks uniquely called by each tool. On the screen, I'm showing you an example of the K562 cell line, which has anti-apoptosis characteristics. Therefore, we expect that the open regions would be associated to genes with anti-apoptosis characteristics and would be enriched for terms such as negative regulation of apoptosis. So on the graph, you can see that only RCL peaks are associated to genes that are enriched for apoptosis process, especially negative regulation of apoptosis process. This suggests that RCL is picking up on good biological signal, even if the peaks are not annotated by other label set or are not called by existing methods. In conclusion, RCL is a novel deep learning framework to identify open chromatin regions with replicates from ATAC-seq data. And the peaks called by RCL are called with higher precision and recall than existing methods. For future direction, we would love to vary the length of the inputs, so alpha is not fixed at 1,000 base pairs. And we can potentially extend and test the framework on other data types, such as ChIP-seq and cut and run data. And currently, we haven't looked at the batch effects or the effects of noisy data when doing contrastive learning together. So that would be also interesting directions in the future. So our paper is online on bioRxiv, and I would love that you would check it out. With that, I want to thank my co-author, Dr. Yudi Zhang, and my advisors, Dr. Geetu Tuteja, and my other advisor, Dr. Karin Dorman. And I would like to thank all the funding resources also. And with that, thanks for your attention, and I would love to take any questions.